Hi, my name is Vivian.
Hi, I'm Rita, and today we're talking about sex linkage and very interesting inheritance patterns.
First, we'd like to offer an introduction. An autosome is any chromosome that is not involved in determining biological sex. In humans and other mammals, biological sex is determined by sex chromosomes. Males have one X chromosome and one Y chromosome, while females have two X chromosomes. So, what exactly is sex linkage? This concept refers to the inheritance and expression patterns of genes located on the sex chromosomes. The X chromosome is much larger than the Y chromosome and contains more genes, which is why most sex-linked traits are associated with the X chromosome.
So, how do we test for sex linkage? A definitive way to test for sex linkage is through reciprocal crosses, where we first cross a male and a female that have different phenotypes. Then we conduct a second cross in which the phenotypes are reversed relative to the sex of the parent in the first cross. In this figure here, we have an example. In the left punted square, a mutant wide-eyed male is being crossed to a wild-type female. And on the right punnet square, we have a wild-type male is being crossed to a mutant wide-eyed female.
Sex linkage also exists in humans. One of these examples would be Duchenne muscular dystrophy, abbreviated as DMD. So this condition happens when there are mutations in the DMD gene, which encode dystrophin, a protein essential for muscle structure and function. Punnett squares can be used to illustrate the inheritance of this condition. Here in cross 1, the female has two mutated X chromosomes, so all her eggs carry the mutated allele. The male has one normal X chromosome and one Y chromosome, so his sperm will carry either X or Y. In terms of results, 50% of offspring will be carrier females and 50% of the offspring will be males with DMD. Since all the offspring inherit a mutated X chromosome from the mother, none will be completely unaffected. Notice that 100% of the male offspring will have DMD.
In cross 2, the male has one mutant X chromosome and one Y chromosome, so his sperm will carry either the mutated X chromosome or the Y chromosome. The female has two normal X chromosomes, so all her eggs will carry the functional X chromosomes. For results, we have 50% of the offspring will be carrier females, meaning that they will have one functional and one mutated X chromosome, and 50% of the offspring will be healthy males. In this case, none of the offspring will have DMD, but females' offspring will be carriers. These Punnett squares illustrate how the inheritance patterns of DMD varies depending on the parental genotypes. DMD is predominantly observed in males, as they require only one mutated X chromosome to express the disorder, while females would need mutations in both of their X chromosomes.
Sex-linked traits deviate from traditional Mendelian inheritance. While Mendel's laws predict independent assortment of alleles on autosomes, sex-linked traits are carried on sex chromosomes. This results in distinct inheritance patterns for males and females, which violate the principle of independent assortment. Now, understanding sex linkage is crucial in diagnosing and managing genetic disorders passed through sex chromosomes, including DMD, hemophilia, and Fragile X syndrome. Genetic counseling and prenatal testing can help predict inheritance risks and guide family planning.
And that's all we got for today. Thank you for listening.
Thank you.